In this video, you will be learning about the citric acid cycle, which is also called the Krebs cycle. This happens in the mitochondria of a cell. Step 1. For step 1, you need 2-pyruvate, 2-coenzyme A, and 2-NADH in order to create acetyl-CoA. The acetyl-CoA is also created in addition to 1 carbon dioxide and 2 NADH. The acetyl-CoA, you need 2, so you have to do that process twice. For this first round of the Krebs cycle, you need 1 acetyl-CoA with 1 oxaloacetate to create the citric acid. This process is catalyzed by citrate synthase, which is an enzyme that speeds up that process. It is important to note that in this process you lose a hydrogen ion, or an electron. That citrate that you created, or citric acid, is then transformed into isocitrate, which is catalyzed by aconitase. In this process, citrate is dehydrated to form cis-aconitate. That cis-aconitate then is hydrated to form the isocitrate, which again is catalyzed by aconitase. That isocitrate then loses a hydrogen ion as well as the NAD, which loses a hydrogen ion to be transformed into NADH. This process releases carbon dioxide and oxalosuccinate. This also loses a hydrogen ion. Because of that, it releases alpha-ketoglutarate, which is important in the next step. For this next step, you need several coenzymes, which include NAD+ and FAD. The NAD+ gets reduced into NADH, and carbon dioxide gets released, as well as succinyl-CoA, which gains a phosphate for GTP. For that GTP, succinate, or succinic acid, gains that phosphate group for GDP in order to create GTP. This is all catalyzed by succinyl-CoA synthetase. The succinate that got released is FAD dependent, which means it must have the FAD, which gets reduced to FADH2. And in that process, fumarate is released. Fumarate is also called fumaric acid. Fumarate is then hydrated to form malate, and that process is catalyzed by fumarase. Malate is also called malic acid. Malic acid is important in the final step, which is NAD dependent. The malate must have NAD in order for the next process to begin. The NAD gets reduced into NADH, which releases oxaloacetate, which, if you recall, was necessary to form citric acid in the very beginning with that first acetyl-CoA that you made. It is important in this step because of this. The oxaloacetate can then be used for the second cycle on the Krebs cycle, which can be used with the second acetyl-CoA that you created in the very beginning. One rotation of the Krebs cycle produces 2 carbon dioxide, 3 NADH, 1 FADH, and 1 GTP. Now, you need 2 GTP, which is why you have to do the whole process again, which is why you needed to get that oxaloacetate released for the second acetyl-CoA. This cycle keeps repeating to create 2 GTP with every 2 cycles. That is the end of the Krebs cycle.